Hi, my name is Adam Carlson. I'm an analyst here at ABG, and it's my pleasure to introduce the penultimate presenter, Peter Van, CEO, and Alireza Biglarnia, CMO of iCoat. iCoat is an unlisted Swedish company in early clinical development for its lead asset, TUM-12, which aims to minimize the damage organs sustain during transplantation and thereby improve patient outcomes, and are initially focused in kidney transplants. I'll pass the word over to you, Peter.
Thank you, Adam. Sound check. Everything is okay?
We hear you.
Great. Okay, good afternoon, everyone. Thank you very much for being here with us today. My name is Peter. I am one of the co-founders and work as the CEO of iCoat Medical, a company founded in 2018 by myself and 5 senior researchers. One of them is with me here today, Professor Alireza Biglarnia, our CMO. If we move forward one page and then another. So what we're going to talk about today are 3 things. I want to give you a brief intro to what we do and who we are, then walk you through our underlying research area and the portfolio we are building, and finally give you a business update of what has happened since we, together with with ABG, led the private placement this spring. So if we go to the next one. iCoat Medical exists as a company because there is a great medical need all over the world for more organs that can be suitable for transplantation. It is not only a worldwide shortage, but it is also a continuously growing waiting list in country after country, and a vast number of people are dying waiting for new organs, mostly kidneys. Just to exemplify the great extent of this need, less than 10% of the global need of organs is met on an annual basis, and each year kidney diseases kills more people than breast or prostate cancer. A driving reason for this is that during a procedure leading up to a transplantation, all organs they experience a various lack of oxygen during the transport between a donor and a recipient. Because of this lack of oxygen during the transplant, a so-called ischemia reperfusion injury occurs, which damages the organ and activates the recipient body's immune system. Our focus is this ischemia reperfusion injury, and what we do is that we will significantly decrease the levels of organ damage that occurs during transplantation with our lead asset, TUM-12. Our firm belief is that this will have 2 significant positive contributions to this industry. Namely, it will increase the life length of grafts, and it will also increase the pool of organs available for patients, since we will be able to use marginal ones that, as of today, are discarded. If we go to the next one. So iCoat Medical as a company, it is a fairly young one, but it is built upon decades of research, which has generated very strong preclinical data, which is our base. This work has taken place by our 5 other founders, all of whom are present in the firm today. And over the past years, we have built a strong complementing team to the founders' respective areas of expertise. We are now 28 colleagues and expect to grow further, both in an R&D side and gradually increasing number of commercial colleagues as well. And what we do together is that we are addressing a very attractive industry segment, namely the organ transplant market. It is non-cyclical, it is global, and the number of transplantations are growing each year. And this is something we do by, in a systematic way, building a portfolio of orphan drug candidates based upon an IP-protected platform. This platform is linked around our work in ischemia reperfusion injuries, and will produce products with multiple indications going forward. The lead one is TUM-12 for kidneys, and we expect to have an approval to commence our first in-human study either here in December or early January. And we have built and continue to drive iCoat based upon 5 academic platforms linked to 5 separate universities: Uppsala, Lund, Linné, SLU, and the University of Tokyo in Japan. These platforms, they give us a great advantage in terms of access to leading research, colleagues, and infrastructure to conduct preclinical and clinical trials and experiments. We expect that in around 2025, we will be ready for market launch with TUM12 for kidneys, and we will continue to advance TUM12 for heart and liver as well. Together, these three indications will cover more than 90% of all transplantations taking place, and a subsequent number of indications will follow. But the total addressable market we will have with these initial indications indicate a value of more than $4 billion a year, and subsequent indications will come as stated. So this is what we do, and if we go to the next one, I'll show you who we are. Next page, yeah, number 6, this one. So as stated, all 6 founders are active in the firm and lead up separate stages of our development and organizational entities. We are also complemented by 3 other colleagues in our management team. David Berglund, serving as our head of strategy and business development. He holds a 10-year background in early stage firms like ours and has a strong track record in transitioning academic projects into commercially viable organizations. Also, Christer Sävholm heads up our firm from a toxicology point of view. He's the former head of toxicology at AstraZeneca and also has a long background at Karolinska Institutet. Finally, Inga Dahlfeldt is our director for clinical trials, coming in with more than 30 years experience and up towards 100 successful trials similar to the one we are about to commence. We are also in the late stage of signing a CFO who will join the management team. He will be publicly announced in a week or 2. But taken together, the management team here comprises a strong track record with complementing experience and expertise, which has a great advantage that I will explain on next page. So if we move to 7. So unlike many other preclinical or early stage clinical firms, iCoat Medical is not derived from a group of scientists with the same background. Instead, we have a complementary area of expertise, and this is important for us because we cover the full development curve from in vitro studies all the way through small and large animal trials into human clinical trials and subsequent commercializations. So in effect, we don't see any operational areas where we are not ready for what lies ahead, but rather a strong organization is here to continue the development of our portfolio. So this is the operational setup that we are very comfortable with. And if we go to the next one, we will cover additional key people. We are very proud of our board of directors, which is being led by Hans Larsson, former chairman of the board for Svenska Handelsbanken, as well as a long operational industrial background throughout Sweden and on an international level for the past 40 years. Also, Bertil Villard, one of the founders of Vinge, supports us, whereas Bo Nilsson and Marianne Jensen-Wern serves as 2 of the founders who represent the core of our work, and also Martin Åmark, our 5th current member. Martin is the CEO of Xbrane Biopharma, a listed generica company which has a long track record of successful partnerships as well as various financing activities. 2 weeks from now, our board will also be complemented by Carl Bjartmar. Carl is the current CEO of Celia Pharma, and for those of you who have interest or experience in the orphan drug industry, he is a familiar name. Recently, he served as the CMO of Wilson Therapeutics, which was a very successful exit to Alexion, now part of AstraZeneca, and constitute the meat and bone, so to speak, of their orphan drug initiative. Finally, we hold 3 key opinion leaders, 1 in each main market that will represent our underlying scientific work. That is Professor Stephen Sachs in Europe, Professor Raya Kandaswamy in the US, and Professor Katsunari Tanabe in Japan, effectively covering all main markets we will primarily focus on. Next one, please. I will now introduce you a bit to our area of expertise as well as focus areas. and that is the ischemia reperfusion injury I've mentioned. IR injury, as it is often called, is a pathophysiological mechanism which leads to so-called thromboinflammation and connected tissue injuries. If I simplify it a bit, the cause of ischemia reperfusion injury can be described as a two-step process, as shown to the left here. First, whenever a lack of oxygen occurs, it leads to a cell injury and changes to the cell's surface. This in turn leads to the immune system being activated and an attack is commenced, which goes towards these altered cells and causes an ischemia reperfusion injury. The ischemia reperfusion injury leads to this thromboinflammation, which can be found in multiple medical areas: cardiac bypass, myocardial infarction, cell transplantation, as well as stroke, and of course, organ transplantation, which is the area where we start, or at least focus for now. And we do this through our product TOOM 12. So if we go to the next page. TOOM 12 is the result of decades of research, and what it does is that it aims to reduce this IR injury. The IR injury, it always occurs when an organ is transplanted. The level of damage and the frequency of it can varies a bit depending on which area of a patient one investigates, but it is always there in every single organ transplantation. In 2012, we proved the mode of action for this product, that it reduces this injury significantly. And how it does this is that it forms a barrier which protects the endothelium and compensates for these damaged glycocalyxes that occurs. So if you look here at the bottom of the slide, this is a simplified illustration of a cell membrane, which in a normal state is attacked by the immune system. But whereas when iCoat Medical's product TOOM 12 is inserted, it acts as a coat that connects to the cell membrane and effectively shields towards these IR injuries. So take the next one. We'll cover a bit more about our product. TOOM 12 as a structure and its function is comprised of two parts, a PEG and a phospholipid. And TOOM 12 works in a way where it interacts with the phospholipid of the cell membranes and shields the surface completely from the immune system's attack. This can be shown in the picture to the left, taken by a microscope, and where a coloring pathogen has been inserted to show this in a luminescent way. Point being, it is an impenetrable coating that covers the organ completely and protects it very, very effectively. And we are confident in this statement due to the preclinical work we have done. So if we go to the next one, please. I want to describe to you the background to the preclinical studies we have done and why we've chosen to do them to a large extent on pigs. Because pigs are very suitable for large animal models for transplantation. And this is because their kidneys are very similar to the human organ in terms of both anatomy and physiology. And furthermore, the studies and transplantation procedures we have conducted, they have been done on pig patients in the same fashion that they would have been done on a human patient. And throughout these studies, we have consistently shown efficacy demonstrating a significant improvement, and the severity of the IR damage has been reduced to a great extent. So we are confident that the results obtained from our pig studies on innate immunity, they have very favorable chances of being able to be transitioned into humans, which we will commence in a month or two as stated. So this is a bit about pigs, and if we go to the next one, I'll talk about pig studies. The pig transplantation models we have conducted are very promising because what we have done is that we've taken two kidneys from the same donor, treated one of them with TOOM 12 and the other one with a control or a placebo product. They have also been done in a compartmentalized environment where all possible actions have been taken to reduce external factors that can have any impact on the outcome, effectively given that the results we have seen are truly linked to the underlying efficacy of TUM12. Go to the next one. I will exemplify this a bit by showing you some results. This is a selection of results from these studies, and to the left you can see that over time we have statistically shown that there has been a significant improved organ function when a kidney has been treated with TUM12 versus the control product. It has also led to a significantly decreased level of general innate immune activities, as shown in the middle, which is further strengthened by a vast selection of biomarkers that is shown to the right, which are completely marvelous in our opinion. The results here, again, they are shown in kidneys, but it is addressing the innate immune system's setup and the consequences of the IR injury we are addressing, meaning that the results you see here, they are not specifically linked to kidneys, but they will transition well into other organs as well. If we go to the next one, we'll show you here what this means for iCoat Medical as a company. We are building our platform based upon our patented work around this IR injury. This platform allows us to develop, as of today, 2 broad product families. First one being called TUM12, that is for cold ischemia and solid organ transplantations, whereas the follow-up TUM20 will cover warm ischemia. We see 3 primary indications for both of them: kidney, heart, and liver, as stated for TUM12, myocardial infarction, cardiopulmonary bypass, and stroke for TUM20. But again, they are based on the same underlying issue and the same type of damage, IR injury, that always occurs in these conditions. We have basically completed all clinical preparations and waiting for the final approval to commence the study on kidneys, and we are rapidly advancing heart and liver as well. If we take the next and final one here, I want to walk you through what we have done since we last time we met some of you earlier this spring. We raised money with ABG, and thereafter we advanced the second product family, TUM20, as well as completed all internal preparations for our upcoming study now. We have filed the clinical trial application to Swedish MPA earlier this year, as well as completed both the tech batch and the GMP batch of our product. We have built a clinical hub and staffed up all necessary resources in Lund and Malmö, and also received the necessary ethical approval for our study. So what lies ahead now is that we are onboarding the CEFO within a week or 2, as well as receiving the team from Läkemedelsverket or Swedish MPA for a clinical inspection in a week. Carl Bjartmar is joining the board of directors, and once the approval has been received, we go into a new phase where patients start to enroll approximately February. Thereafter, a number of corporate initiatives and restructurings will take place in order to prepare for our expected IPO, which will occur late Q4 2022. And the main purpose of the IPO is to finance the subsequent phase 2 study, which will be a large multinational, multi-center study. All in all, iCoat Medical continues to execute on our business plan. We are very happy and thankful for our current investors who support us, and we are moving forward according to plan towards the IPO by the end of 2022. And before that, I hope to be able to come back to all of you, either individually or in group, to present the company to a greater extent. But for now, I think our time is up here. So I will hand it over to Adam and anyone else on the line who wishes to provide any questions or comments. Thank you all.
Perfect. Thank you very much for that presentation, Peder. First question, if I could, just on the market that you're addressing initially, the kidney transplant market. What are we seeing there in terms of trends for the kidney transplant waiting lists in the major markets? How are those evolving? And then separately, if we look at the kind of completed transplants, deceased versus living donors, what's that split like, and how is that expected to evolve over time? I'd be interested to hear.
So the transplant market itself, as with most indications, the US is often driving initiatives. There we see several ones where they try to get the number of transplantations up. It's also an area which is coming under scrutiny, not only because a vast number of organs are being discarded, but also because prices are soaring. We are in a currently early stages of an in-depth commercial assessment how this will impact our commercial preparations. But regardless, we will target the US as the primary market. And that is not only from a commercial point of view, but we see several of the large transplantation centers there, which will be suitable partners for our upcoming study. I want to come back to you in a few months and say how we will target Japan and Europe, but we will go for the big five and the Nordic countries where we see the number of transplantations going up, as well as, I don't want to say great advancement, but at least indication of advancement among involved stakeholders for how to get the number of transplantations up, as well as reducing the obstacles to get approval for new innovative products. With regards to the splits between living and dead donors and what is developing there, I think I should pass it over to you, Alireza. You're on mute.
All was the same. Concerning the living and deceased donor, what is important to know is that what we actually try to do is to develop a treatment that deals with inflammation that is induced by ischemia or deprivation of oxygen. The deprivation of oxygen can also happen in living donors. So, and this is also very obvious with these developing programs in which we send kidneys, even from living donors across borders and regions, which means that the ischemia time is increasing in these patients. What the development, in the clinics, we really try to do everything we can to increase or optimize the organ availability, to utilize them well, and also to allocate donors, both living and deceased donors, as good as we can. But the reality is that the demand will always be greater than the availability of organs.
Okay, great. Thank you both for that. Another question, if I could, on the preclinical data you've generated so far in pigs, and we saw some of that in the presentation. I was wondering if you could comment on kind of how predictive the kind of kidney function and inflammation markers that you're measuring in the first few days post-transplant are for the kind of long-term outcomes of a kidney transplant. And I guess as a follow-on to that, the kind of early interim 6-month data or so that you'd be generating from the phase 1/2 study, how much can we read out of that? How predictive will that be of long-term organ outcomes? And I guess how much would that de-risk the continued clinical development?
That is a great question. What we know is that ischemia that leads to inflammation very often cause the kidneys to function badly in a way that we'll really need to apply dialysis for this patient during the first weeks. And this is a situation which is, or this is a clinical situation which is called delayed graft function. And we know that delayed graft function has a very vast impact on outcome, both short-term outcome and long-term outcome. When we implement treatment that potentially decrease the incidence of delayed graft function and maximize the function of the kidney, it's very logical to assume that you also has a positive impact on short and long-term outcomes. In terms of biomarkers, What is interesting to know is that the same inflammation we have seen in the pig models, you can also see in the humans. So you really can replicate this kind of inflammation in the same way you can do in a pig transplant model. And this is the coming study that we initiate. It is a first-in-human study, and the sole purpose of this study, and this study is designed only to show safety. However, we have implemented also biomarkers, and these are biomarkers for inflammation, which we know that are that works in humans, and we try to screen for this inflammation and see the effect. And hopefully, we will also have not only safety endpoints, but also some efficacy markers that would help us to understand more the pathogenesis in patients, in transplant patients.
Okay, great. I think we're almost out of time. I might ask one final short question, if I could. And I don't know how easy it is to answer in a short way, but in terms of the clinical kind of development risk, ideally with any clinical study, you want to minimize the factors beyond the treatment itself that can kind of influence the outcomes. Here, by definition, there's obviously patient-related factors, organ-related factors, the skill of the surgeon, and so on. How should one think about kind of these factors kind of influencing the outcome once you move on to kind of larger efficacy-based studies? What are your thoughts there?
That is a problem with all clinical trials, because, as you mentioned, the bias and the virality of bias are pretty much high. The way you try to counteract that is by designing the study in a way that it gives the highest evidence possible and the best control group possible. And that is the design which we have chosen for the first-in-human trial, which is a randomized, double-blinded, placebo-controlled study. And the same way we will also proceed with the upcoming efficacy study. As Peter mentioned, this study will be done in a large number of patients, and the number of patients can compensate for these biases. And to do that, we need to include many centers across the countries, both in Europe and the US.
Okay, perfect. We're all out of time. Thank you very much, Peder and Alireza, for an interesting presentation and Q&A.
Thank you very much.
Thank you very much. Have a nice
Bye.
day.
And you.
Thank you all.